I will be sharing with you. So, Shilin, I just want to double check. Can you see my screen?
Um, yes, I can see your screen.
Okay. So I want to first apologize to the audience that I had trouble do the presentation mode of my slides. So this is the best I can get. But luckily, my slides are with big fonts, so I hope you can read my slides okay. All right. So I always start with a slide talking about drug development process. Um, the way I believe those finding is important is because I believe for every molecule, um, if that molecule is effective, is efficacious, that molecule must have changed our biological system. If it changes our system, it has to be toxic. Uh, so uh, we know if it does not change our system, if this molecule does not change our biological system, it's something we call placebo. All right. So if it works, it must be toxic because it changes our system. So we know every drug is toxic. And then how come there's so many drugs on the market? It's because for every drug, there's a non-toxic dose. So the way I view the entire drug development is about finding the right dose for that molecule. So let's start from drug discovery. All right. At, at the discovery stage, um, I think the technology are changing nowadays, but I entered in the business back in 1987. In the old days, the scientists they did this. They come up with some chemical, and they thought the chemical could work on this tissue. So they put in the petri dish with the highest concentration, and then they pour half of that concentration high concentration material to the next petri dish and fill up with water, and then pour half into the next. So the concentration starts with 100%, then 50%, 25%, so on and so forth. And then you have all these petri dishes, and they have the tissue. They dip the tissue into it and measure the response. And then you're going to have a what they call concentration response relationship. So you can take the log scale of all these different concentration. You line up them from low to high, and then you look at the response at the very low dose or very low concentration. There's no response. It has to increase somewhere in order for the compound to move to the next step of development. All right. So if you keep on increasing the concentration and there's no response, the response is not changing, then you know you don't have a molecule. So you move on to try to find the next molecule. So that's from discovery. You have to look at this concentration response relationship. And then you move into a non-clinical development. So non-clinical development are the development of this drug outside of live human body. So the discovery is trying to come up with a new compound, but the development is trying to observe what the compound does to see if the compound is good or bad. If the compound is no good, then we stop developing it. So you can imagine that discovery, their job is keep coming up with new compound, and the development, both non-clinical development, is trying to select out the ones that doesn't work or having toxicity or having problems formulation, stuff like that. So in non-clinical development, one of the important milestone is what they call toxicology. So in toxicology, they have to look at three dimensions of this drug. They're looking at concentration, species, and duration. So they have acute, subacute, subchronic, chronic kind of different lengths in time, and they try different species. In the concentration, they can start with a 0.001 to 0.003 to 0.01, 0.03, 0.1, 0.3, 1, 3, 10, 30, 100, and so on and so forth. And the unit is what they call MPK, it's milligram per kilogram. So they're looking at the concentration of the test drug and the body weight of the animal. So it's milligram per kilogram. So they increase when they see some toxicity, they will stop. Now, So they try maybe one or two species at the beginning. All right. So you go through non-clinical development. They're trying to look at different concentration response relationship. One of the deliverables from toxicology is what they call NOAEL, NOAEL, which is non-observed adverse event level. All right. So by increasing the concentration up to a point, they did not see any toxicity. They did not see any problem. But beyond that concentration, there's some problem. Okay. So the highest concentration that does not cause a problem is called NOAEL. Remember, this is in terms of milligram per kilogram. So they have this kind of uh, sort of concentration response relationship. And now, if the drug is good, if the molecule passed the drug discovery and the non-clinical development, they'll eventually get into human being. Once you're in human body, there are phase one, phase two, phase three, and phase four. So the way to under phase one, two, three, and four is as follows. At phase one, you're starting with the first time in humans, so you're not going to go with very high dose. That's why this NOEL is very important. So the toxicology group will recommend this NOEL, and you go through what they call a translational medicine. You translate that concentration into human body. So after that translation, that NOEL translated into human body, we call it HED. It's a human equivalent dose. So you have that HED, and then you further multiply by a safety factor like 1/10 or 1/20. So you want to make sure it's low enough. And once you phase one, you're escalating. So you're increasing dose from low to high. So what does it mean by high? The upper limit of the dose range once you're in human body is what they call a maximally tolerable dose. So you push the doses up that the human body can still tolerate. And beyond that maximum tolerable dose, which we call MTD, then human may not be able to tolerate. So it's maximally tolerable dose, MTD, is going to be the upper limit of the dose range. So what phase one has to achieve are MTD and PKPD. So what's PKPD? Pharmacokinetics and pharmacodynamics. So pharmacokinetics is what does the drug do to human body? And pharmacodynamic is what does no pharmacokinetics is what does the human body do to the drug, and the pharmacodynamics is what does the drug do to human body. Of course, we develop a drug is we want to achieve the PD effect, but before we study PD, we need to know the PK. What does the body do to the drug? Okay, and then um, you escalate up to MTD. Uh, because you want to study PK/PD at phase one, uh, they tend to, for chronic disease, they tend to use healthy normal volunteers because for patients, their uh, metabolism may be affected by the disease. And the other issue is that if the patient with some kind of disease, that patient must take some medication to control the disease. The medication the patient's taken could interact with our study drug that changes the metabolism system. Okay, so you want to start with healthy volunteers, normal healthy volunteers for chronic disease. You start from phase one, and you escalate up to MTD. Now, after you deliver these information, PK, PD, and MTD, you engage into phase two trials. Once you get in phase two trial, you want to lower the dose from the MTD down to the minimum effective dose. So in phase two, the two considerations are proof of concept and dose ranging. So phase two, you want to first be able to show the drug works. So that's what we call proof of concept. If you can show the drug is efficacious, then you move down to dose ranging. Now, the reason why we want to do a proof of concept at phase two is remember at phase one we use healthy normal volunteers, so from phase one you don't get to see drug efficacy. So phase two is the first chance you get to see efficacy. If the drug doesn't work, then you're done. So if the concept is proven, you move on. The next step is dose ranging. So phase one is PK/PD and MTD. Phase two is um, POC and dose ranging. And now you have the upper limit is MTD. The lower limit of the dose range is minimum effect dose. So upper limit came from phase one, lower limit came from phase two. They come up with this range of doses that the drug is efficacious and safe. And from this range, you select one or few doses. You go to phase three with long-term and large-scale confirmatory trial to confirm the doses you select in phase three are both efficacious and safe, and then FDA could approve. Once the drug is approved after phase three, then you engage in phase four, which is post-market. Post-market being after approval, and there are many drugs after approval, they're still making those adjustments. Some are just higher, some are just lower. So these are the phases one, two, three, and four. Is all about finding the right range of doses. So starting from discovery until post-marketing, in the entire lifespan of any molecule, is about finding the right range of doses. That if it's too high, it's toxic. If it's too low, it doesn't work. You try to find the range so that it is efficacious and safe. All right. Uh, so I mentioned about chronic diseases. So we use healthy volunteers. All right. So for non-life-threatening diseases, you start with healthy normal volunteers, and you're looking at PK properties being pharmacokinetics. What does the body do to the study drug? And help recommend dosing frequency and try to escalate the doses up to maximum tolerable dose. And dose escalation design or crossover design are popular in phase one. All right. So dose escalation design starts like this. You start with a group of healthy normal volunteers. You expose them with the lowest dose. That's where the novel you translate into HED, you multiply by a safety factor at a very low, very safe dose, and you see if there's any problems, side effects, or any issues. If there's no problem, then you recruit another group of subjects, no more healthy volunteers, at a higher dose. And if there's no problem, you keep on increase until you can get to the MTD. In this process, there's things they call SRD or MRD, single rising dose or multiple rising dose. So single rising dose is you have a group of cohort and you look at there's any adverse events or any problems, but these subjects just take one dose. All right, just dose once and you follow them. The multiple rising dose is that they're taking multiple times of the same dose strengths. So this multiple rising dose is not changing dose strengths, it's changing the number of times the patient or the subject is being dosed. Okay, so that's SRD and MRD. You try to escalate up to this maximum tolerable dose. So phase one has to deliver this maximum tolerable dose and PK/PD. So why does phase one have to deliver these information? All right, in phase two, we're recruiting patients, right? So these patients come in to the clinic, and we call them outpatients. They're not being hospitalized. They just go to the uh, clinic, go to the doctor's office, and the doctor will do the evaluation and dispense a study drug. They can take it back home and take them. And these outpatient visits, we have to tell them how often they have to be dosed every day, right? Twice a day, three times a day, and then how often that dosing frequency came from PK information. So later I'm going to talk about the PK. There's a time concentration curve. You try to find out how long does the drug stay in the body and for how much. So PK/PD will help to guide the dosing frequency. Why is MTD important? Okay. Remember I said phase two, the first phase two trial is a proof of concept trial. Is this is the first opportunity you get to see drug efficacy because before phase two, all of the preclinical studies and phase one studies, we never get to see whether this drug works in human patients or not. So phase two is the first time you're exposing this drug to patients, and that's the first time you get to study drug efficacy or clinical efficacy. Because before the first phase 2 trial, you probably have PT markers, you probably have animal models, but you don't know whether the drug really works in patients or not. So phase 2 is the first opportunity that we get to see whether the drug works or not. So you want to do a POC. So the POC typically was two treatment groups, test drug against placebo. And if the drug does not work, then you're done. You say, okay, all the previous effort that we came up with the compound, this compound does not work. If the drug works, they move on to dose ranging and possibly phase 3 trials. So this POC is a go, no go decision. Should we continue to develop the drug or should we stop? Now, if that's the case, the test drug against placebo, what dose of the test drug are we going to choose? Are you going to choose high dose or low dose? Let's think about it. If at the low dose against placebo, the drug doesn't work, am I comfortable to say the concept is not proven? No, I'm not, because a higher dose may work, right? So I want to go with the highest possible dose. If the MTD, the maximum tolerable dose, and placebo, I still cannot differentiate. I mean, there's no difference from placebo between placebo and the highest dose of test drug. Then I'm comfortable to say, well, the drug doesn't work, and we're done with it. All right, so that's why we had to find the MTD from phase 1. Uh, in reality, in practice, there are times the phase 1 may deliver MTD incorrectly. We have situations where the MTD was underestimated or overestimated, and both are huge problems. Okay, now let's talk about what if MTD is overestimated. So if the true MTD is 100 milligram and the phase 1 tell me is 200 milligram, so I start with my POC being 200 milligram against placebo. Then what happened? I may see a lot of adverse events because the true MTD should be 100, and now I'm exposing these patients with 200 milligrams, so they're going to develop adverse events. But overestimation of MTD is less a problem than underestimate MTD. All right. If for underestimation, if the true MTD is 200 milligram and the phase 1 tells me it's 100 milligram, then what I do is I do a POC of 100 against placebo. Right. So I designed the study 100 in placebo, and by the end, 100 works and very safe. And you think the efficacy is not good enough, and you think a higher dose may deliver better efficacy. So then what you do, you design another study, 150 against placebo. And after the result came out, 150 works better than 100. It's still very safe. It looks like there's still um, space you can improve on efficacy. So you want to design a 200 against placebo. So what's wrong with the picture? All right. Remember, for new drugs we're developing, it's under patent protection. The patent life is only 20 years. For a POC study, from design until decision making is easily half a year. So if you have to design a couple POC, it's eroding your patent life very badly. So either under or overestimate of MTD could be a problem. But in reality, do we really know whether MTD is underestimate or overestimate? We never know that. Okay, we can only take the phase 1 information as if we know MTD. Okay. All right. So help recommend dosing frequency as in MTD and dose escalation design are popular. All right. Now for life-threatening disease, that's different. For cancer, all right. So the cancer is this. The traditional cancer treatment is like this. All right. We we diagnose this patient with cancer. The first thing is to remove the cancer, remove the tumor. All right. So you can either surgical operation to cut out these tumor tissues. Or sometimes you cannot use surgical operations, you can use radiation. So you can radiation to get rid of the tumors. So the first step is to get rid of the tumor, whether it's operation or radiation. And after you get rid of the tumors, you still concerned there may be remaining tumor cells in your body. Then you follow with chemotherapy. All right. So what does chemotherapy do? chemotherapy is trying to use that to kill off the remaining tumor cells. So these chemotherapy are known as cytotoxic. It's toxic because the efficacy of these drugs, of these agents, is because they can kill the tumor cells. And we can imagine if these agents they can kill tumor cells, they can kill normal cells also. So they're toxic in nature. So for chemotherapy, it's toxic already to begin with. So it's no longer ethical to recruit healthy normal volunteers into phase one. So cancer trials, you start with patients in phase one. So who are the patients that are going to participate in phase one trials? It's typically the cancer patients, they tried many other drugs and it doesn't work for them. This phase one trial may be their last hope. So they participate in the trial. So you can imagine these cancer patients, they may not have the tumor type the drug is developed for. So although you're developing a drug for lung cancer, but the participant may have a neck cancer or probably have a kidney cancer or something. So you still cannot get to see the drug efficacy, but at least you can do this dose escalation to help understand where the MTD is, the maximum tolerable dose. All right. So for cancer drugs may not be ethical to use. Placebo control, of course, is toxic, and you have patients with cancer, you cannot use placebo control. All right? May not be ethical to recruit normal healthy volunteers, open label, single arm, dose escalation study designs. All right. In oncology, you start with cancer patients, and it's not ethical to do placebo control. You try to define something they call DLT, dose limiting toxicity. And you try to develop this drug such that the MTD is somewhere, the toxicity at the MTD is controlled under gamma. This gamma is a probability, probably one third or one sixth or something like that. So gamma is a target probability of toxicity. All right, so traditionally we have this 3+3 design. I think I can show you this figure. All right, that makes things easier. All right, so you start with 3 cancer patients, and if none of them have any DLT, dose limiting toxicity, you'll escalate to the next higher dose. Right. So for this dose, if there's no adverse events or no DLT, you'll escalate. So whenever you see a DLT, all right, if there's one DLT, you add 3 subjects at the same dose. All right. So if among the 6 patients there's only one DLT, then you escalate. But whenever you have 2 or more DLT, you know the MTD is exceeded. So the dose lower than that dose is what called MTD, maximum tolerable dose. Okay. All right. So that's pretty much a brief introduction about phase one. So the 2 deliverables for phase one are PK, PD, and MTD. And now you engage into phase two clinical trials. All right. Uh, The first phase two is proof concept, followed by dose ranging. All right. And the objective is to propose dose or doses for phase three design. Okay. You try to move the doses down to minimum effect dose. All right. So a typical dose ranging study looks like this: high dose, medium dose, low dose, and placebo. You got 4 treatment groups. All right. Placebo, low, medium, high. You have 4 treatment groups, and you try to see if there's any dose response relationship, with an objective of moving the doses down to minimum effect dose. Hopefully, we can find the minimum effect dose. If you can find the minimum effect dose, then you can establish the lower limit of those dose range. Remember, MTD came from phase one, so maximum tolerable dose, which is the upper limit of your dose range, and now you move down to the lower limit of dose range. Well, keep in mind, moving doses up, the limiting factors are safety or DLT. All right. If there's adverse events or toxicity, you have to stop. Moving doses down is based on efficacy. All right. So how do we establish efficacy? Because from a hypothesis testing point of view, we're trying to separate the two distributions, right? The null hypothesis says mu t equal to mu p, that means the test drug mean and placebo mean are not distinguishable, right? You have two distributions. I have a distribution from placebo, I have a distribution from test drug. And if these two distributions are not distinguishable, I accept null. I say the drug doesn't work. All right. Now, if you can separate the two distributions, that the test drug and the placebo, they're very far apart, then you say I reject null, right? So how far apart? If your observed p-value is less than alpha, and you say statistically significant and reject null, and you claim there's efficacy. Okay. So moving doses up and moving doses down, the entire considerations are different. Moving doses up, you watch for toxicity. Moving doses down, you try to see if you can separate the two distributions. Okay, and hopefully you can find mean effect dose. Later, I'm going to explain why mean effect dose is mission impossible. Okay, among all the drugs approved on the market, there's not a single drug I know clearly what the mean effect dose is, because finding mean effect dose is very, very difficult. All right. Now, if the dose range is not found in phase two, it'll be too expensive to later phases. So I give you a example here. All right. So this company, they try to market this drug. Uh, they got the approval in many countries, not in U.S. All right, and they try to bring the drug into U.S. for approval. Now, drug pricing in most countries, they are linear to dose. So if 100 milligram is $1, 200 milligrams is $2, 300 milligrams is $3. So drug price is linear to dose. So this company, they have this drug approved in many countries at 100 milligram. Now they try to bring to FDA. So FDA look at the submission, says, uh, well, it looks good, but we want to see a dose response below 100 milligram. So this company went ahead and designed a dose ranging trial. Well, 100, 50, 25, and placebo. Makes sense, right? 100, 50, 25, and placebo. And after the study came out, 25 milligram worked. FDA says, okay, looks good. We'll approve at 25 milligram. Now, what's the consequence? Every other country is trying to lower to 25 milligram. And the company's revenue just lost by 75%. Okay. So it will be too expensive to find the right doses in phase three or phase four. And when I tell the story, people say, Naitee, you claim you're a scientist. You were talking about the statistical science to help with drug development. And why do you talk about the marketing commercial issue? I say, wait a minute. This is absolutely a scientific problem. Why? Because the only chance you can find mean effect dose, you can find the range of doses is phase two. If you miss phase two, it'll be very expensive in phase three and phase four. It's a hard lesson we learned over the years. All right. So what's POC? Typically, two treatment groups, parallel design, placebo controlled. And the test drug is at MTD or dose close to MTD. You want a very high dose, the highest possible dose that we try to differentiate from placebo. If this highest dose cannot differentiate from placebo, then we say we're done. We're not going to spend any resources on this molecule. But if concept is proven, then we're going to gear up. This drug may go to phase three. Okay, so the POC is about a go no go decision. If the drug doesn't work, it's no go. If the drug works, it's going to be a go. All right. Usually, short-term clinical efficacy endpoint and moderate sample size. I want to emphasize this moderate sample size issue. All right. Now, this is early phase two. All right. All the data we have up to this point in time are preclinical and phase one. We have no phase two data yet. All right. So we still don't know whether the drug works or not. From that perspective, it may not be ethical to recruit a large number of patients to participate in the trial. You still don't know the drug works or not. And from the investment point of view, the company, well, the big company usually have multiple drugs being developed. Now, this drug has not go through POC. We don't really know the drug works or not. Why should we invest a lot of money on this drug? So your sample size is rather limited in a POC study. All right. So let's move on. All right. I mean, we're statisticians. We're familiar with sample size calculation. All right. And for most part of my career, um, I thought sample size is part of a study design, which is correct. But the more I'm in this industry, the more I'm convinced sample size is the study design. In other words, sample size reflect the bottom spirit of this entire study design. Naitee, why would you say that? All right. So we know sample size n is a function of four quantities, alpha, beta, delta, sigma. So let's think about what are these four quantities. What's alpha? So of course we know alpha is a probability reject a null hypothesis. Reject a true null hypothesis, that's alpha. But actually, when you communicate with the physicians, with the team members, you don't say that. You say alpha is the risk of developing a placebo. What does that mean? All right. The underlying truth is the drug doesn't work, but purely by chance, you observe a p-value less than alpha, and you think the drug works. So you made a go decision, but that was a wrong go decision because it's truly a placebo, right? We actually saw that in practice. We saw some of the drugs we tried to develop eventually proven was a type 1 error. Okay, it actually happened. All right. So alpha is the risk of developing a placebo. So beta is a risk of giving up a good drug. So alpha and beta reflects the risk the team is willing to take. All right. Now, what is delta? What is sigma? All right. Delta is a treatment difference. Remember, this is the first um, phase 2 trial, and we have never seen drug efficacy. So we're designing this trial assuming the drug is efficacious, but we never know what the true delta would be. And sigma being the within group standard deviation. Okay. Now, how do you find the standard deviation? We look at published materials and we look at drugs of the same class, and we may guess this sigma. All right. So among the four quantities, alpha, beta, delta, sigma, the easiest quantity is sigma, and the most difficult quantity is delta. So how much treatment efficacy does this drug deliver? We don't know. All right. So now what happened is the the physician or the trial monitor, whoever is in charge, the leader or the manager, came to my office. It's Naitee. Um, we need to design a POC trial, and can you do a sample size for me? Okay. So this is the opportunity for me to make contribution to study design. And I do think statisticians or statistics as a profession is probably the most important science across the entire drug discovery and development process. In a way, it's not in an absolute way, but in a relative way of looking at statistical science, it's probably the most objective science. Because starting from discovery to non-clinical development to phase 1, 2, 3, 4 clinical development, every other profession, they have very strong level of subjectivity. Even statistics, we have subjectivity in it, all right? But when we choose a statistical method or something like it's subjective. But we're trying to stay as most objective as possible. And sample size is really reflecting how we communicate the risks and the assumptions we're dealing with this molecule under development. So I mentioned sigma is the easiest quantity. You can just by reading publications or getting approved drugs from other drugs of the same class or something like that. So sigma is easy. The most difficult part is delta. You don't know how big a delta the drug will deliver, how much a difference between test drug and placebo. So you have to come up with an assumption. So I'm assuming in the next few slides that delta is positive. In other words, the more positive, the better the drug is. All right. And assuming sigma equal 1, because you can use delta divided by sigma, and then your variance become 1. So if you have these quantities, delta and sigma, and then sample size can be calculated given alpha and beta. All right. Now, if the full quantities where alpha and beta are the risk the team is willing to take, and the delta and sigma are the assumptions the team is willing to make, is there anything more important than this design? You in the study that in the entire study design, sample size reflects the risk in the assumptions. So sample size, the more I'm in the industry, the more I'm convinced sample size is the most important part of any study design. Okay. Now, proof of concept by nature is hypothesis testing. All right. We have two distributions. So we have the placebo distribution, we have the test drug distribution. We want to see if we can separate these two distributions. Is hypothesis test framework. No hypothesis says the two distributions are not distinguishable. Alternative hypothesis says they're very different. All right, how different? The p is less than alpha. So what does it mean by p less than alpha? So from drug approval point of view, FDA can never say this drug works. But here's what they can say. They can say the probability the drug doesn't work is controlled under alpha. So if p is less than alpha, then the truth of the matter is the drug may still do not work. The drug doesn't have any efficacy. But because p of less than alpha, we reject the null. But if the truth is the drug doesn't work and we still reject the null, that probability is less than alpha. That's the way we understand this false positive rate, the risk of developing a placebo. So we try to communicate with the non-statisticians of what we understand. This p less than alpha really means you're taking a risk of making a wrong decision. If you make a good decision because the p is significant, be careful. There's still a big probability the drug doesn't work. We just don't know. Okay. All right. So it's hypothesis testing framework, and primary endpoint is clinical efficacy, and pre-specified two-sided alpha could be greater than 0.05. Okay, I designed POC study with alpha being 0.2. And if you look at publications, there are studies designed with alpha being 0.4. Wait a minute, alpha being 0.4. Flip a coin is 0.5. How can you tolerate such a high risk of making a wrong decision? Yes. All right. So why do we have to design study with such a big alpha? Well, you can imagine the entire drug development process is a huge gambling process. You invest some resources, you try to get some data. And if the data tells you the drug is good, then you do more investment, you buy the next set of data, and so on and so forth. It's just like you at the poker table. You put down some chips, you buy a card. All right. If the card looks good, you put down more chips, you buy more cards, right? It's step by step of a go no go decision, right? Because if it looks good, you're going to put down more chips. If the car doesn't look good, you're done, right? So at every step of drug development, you're making that go no go decision. And this decision is most critical in the POC stage, the first phase 2 study, proof of concept. And at this point in time, you have a small chunk of chips. You don't have a huge amount of chips. And you come to Naitee, you ask me to do a sample size. I cannot say your chip is too small. You're going to have too little sample size. I cannot do the study. I cannot say that. So I don't do that. I say, okay, this is how much chip you have for the gamble. Let me help you get onto the table. All right. Now, look, that P the alpha being zero point 0.2. What does it mean by alpha being zero point 0.2? If by the end I got p-value less than p-value of 0.19, I say statistically significant. If the p-value is 0.21, I say statistically not significant, right? But that doesn't mean I said anything meaningful or useful or informative, right? But can you imagine for that study, what kind of results did I get? Yes, I designed the study with alpha being zero point 0.2. By the end of the study, I got p less than 0.0001. The conclusion is very clear. It's a goal decision. So you really have to think this as a gambling process. All right. You don't know whether the drug is good or bad, but you have to bet with the chips you have on hand to bet. So you end up with a small sample size. That doesn't mean your drug is no good. It means, in my example, they have very few chips, but they got an ace, very good card. Now, but what's the basic problem of small sample size is you lose precision. In most cases, if the drug is not that good, you cannot make a decision. Remember, the deliverable of a POC study is a go or no go decision. If you cannot deliver this decision, this trial failed because the trial cannot tell you what decision to make. So that's a problem of inconclusiveness. So when we design trials, statisticians only think about alpha and beta. And not many people talk about the risk of inconclusiveness. All right. Now, with given alpha or beta, you design a trial and they make clear decision of go or no go. It's easy because they make a go decision at most alpha probability they're making a mistake. If they make a no go decision, at most a beta probability they're making a wrong decision. But what if they cannot make a decision? And that can be expensive also, right? I give you this MTD underestimation issue. You have to redesign another study, and that delayed your patent life. Okay, so when you design a POC study, you have always have to have this in mind. We have to help with a clear go no go decision. All right, so the success is not a go decision. The success is not a no go decision. The success of a POC trial is you can make a decision. Okay, so that's alpha and beta, and we want to come up with a clear go no go decision. So here I'm going to propose a communication tool as to how a statistician communicate our understanding to the non-statisticians, including the physicians, the upper management, the team members, whoever need to understand the critical part of the thinking of designing this POC. Okay, I forgot whether I said this or not. I said POC looks like simple, right? Only two treatment group. It's very, very, very difficult. And I'm going to explain to you why it's so difficult. All right, first, we'll talk about probability. You can flip a coin. All right, if I flip a coin, I say the probability of getting a head is 1/2. The probability of getting a tail is a half. That's fine. All right, so I flip a coin. Now the coin's landed. Is there any probability? There's no probability. You open up and you see whether it's a head or tail. There's no probability. In clinical trial, it's like flipping a coin. You're looking at the probability, right? But once the coin is landed, there's no probability. All right. So in clinical trial, when did the coin land? When you unblind the trial. All right. So when you complete the study, you say, okay, the study is completed. Let's unblind. Once unblind, the coin's landed and there's no probability. So the reality is, after unblinding, there's the results came out. And everybody have a say. Somebody says it's a wonderful drug. Somebody says, well, it's mediocre. And different people have different opinions, including the physicians, the upper management, the team players, all but everybody have their opinion. When that happens, as a statistician, I become a pair of hands. Naitee, do this analysis. Naitee, do that analysis. I become a pair of hands. I just do the work. But before unblinding, we're talking about probability. I can communicate with the team so that they understand the true value I can deliver as a statistician because we are the thinkers of a clinical trial team. We are the brain of the team. Yeah. Yes, I will do analysis. I'll be a pair of hands. But before being a pair of hands, I have to think. I have to be the brain of the team. And how can you show you're the brain of the team? You have to communicate. And that's why I'm going to give you a communication tool here. A communication tool is proposed to help the team members in understanding the risks. All right. And the timing of the discussion has to happen before breaking blind. So before breaking blind, I'm the brain of the team. After the blind is broken, I'm just a pair of hands. Okay. And after design is finalized and clear go no go criteria can be documented. Okay. So from study design point of view, all right, let me give you an example. All right. So this team leader or project manager or trial physician, whoever is leading this trial, came to my office. Says, Naitee, we're going to design a POC trial, and can you calculate sample size for me? I say, yes, I'm happy to. So at this point in time, that's my opportunity. Remember, that's the opportunity for me to contribute to the study design. All right. I have to sidetrack just a little bit. We are statisticians. We're trained with statistics, statistical thinking, and we're good. Fine. But do you know how the physicians think of us statisticians? All right. Over the years, I work with many physicians. We became very close friends. And they told me how they view us. All right. One physician told me, says, Naitee, do you know why I went to medical school? I say, medicine is a very good profession. You are helping people, you're saving lives and stuff like this. Is no. Naitee, my math is not good. And he told me, you must be a mathematical genius. So I had to tell him it's totally misunderstanding. I had to explain, listen, the reason I try to work with you, try to explain things clearly is because I'm a very bad mathematician because my math is awful. Okay, I don't have a good math training, and I can sympathize with people who do not have good math. So that's why I try to explain math or statistics in a way that everybody could understand. All right. So in general, physicians think of statisticians as mathematics genius. They're nerds. They don't know how to communicate. In fact, we going through all the training, we never learn how to communicate. If you go to business school, you have to do case studies, you have to do group discussions. If you go to like other science like physics, chemistry, or biology, you have to perform experiment. Lab experiment. You have to have your lab partners. You have to communicate. But statisticians, all our training are the professor writing on the board, and you take notes, and the homework, you go back, close your door, and try to prove theorems. Okay, so we don't have enough training of communicating. So communication is absolutely key. All right, so let me back to this design situation. So this clinician came to my office, says, Naitee, we need to design this POC. So of course I have to ask about the primary efficacy endpoint. I need to understand the disease and all that. And then I say, calculate sample size, I need alpha, beta, delta, sigma. So we discussed alpha, beta, delta, sigma, and everything is good. Before he leaves my office, it's 90. Oh, by the way, we're limited to only 100 patients. So sample size is limited to only 100. I say, yes, I can do that for you. All right. So after the physician left, I look at the alpha, beta, delta, sigma, plug in the software. I need 500 patients. So I went back to the office. I said, well, based on our discussion, I need 500. 500, that's too many. No way. We can only afford 100. I said, fine. let's change our assumption, right? To make it feasible with 100 patients. Okay, so how do you change the assumptions? Well, the most effective way is to increase delta. All right, you can increase a tiny delta, you can reduce a large sample size. So just keep increasing delta. I say, can the delta be like this? So push, 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 until he said, oh, wait a minute, that's not humanly possible. The delta can at most be this big. I say, fine. So we came up with this delta. This is the delta I can calculate sample size, and I came back with I still need 300 patients. Oh, 90, we can only afford 100 patients. So I say, fine, let's increase alpha, increase beta. That's why I came up with that design with alpha being zero point 0.2. So we know in order to make the study feasible, when we have very limited chips, we have to increase delta, increase alpha, increase beta to make the study doable. Okay, so that was the background thinking process. All right, so now I need to communicate with the team about how do you make your go no go decision. So given alpha beta delta sigma, we came up with a sample size. So we had to discuss before breaking blind, because after breaking blind, I'm useless. I'm just pair of hands. I'll just do analysis from left to right. But before unblinding, that's the opportunity I can communicate these risks. Okay. And I say after study design is finalized, because if I go through this communication tool and they understand what I'm talking about, they may change the design, they may increase sample size. There's no need to increase sample size because for this given sample size, we're willing to take these kind of risks. Sometimes the upper management told me, says, Naitee, we hire you statisticians try to help us reduce the risk. I said, wait a minute. I cannot reduce the risk. All right. If you want to reduce risk, increase sample size. Your risk is going to be decreased, right? But you have only limited sample size. I said, I'm here to help you quantify the risk. Given the sample size, what is the probability you're going to make a type 1 error? What is the probability you're going to make a type 2 error? With a feasible study design, with only 100 patients, and with these kind of alpha, beta, delta, sigma, and that reflects this design, that reflects the risks the team is willing to take in terms of making wrong decisions and the assumptions the team is willing to make. Here's a delta, here's a sigma. So given these assumptions and risks, I came up with a sample size. So the bottom line, sample size is the fundamental spirit of this entire study design. All right. So I prepared the next communication tool, and the timing of discussion is before breaking blind, after the design is finalized, and we had to come up with a clear go no go criteria. So here's what we did. Okay. So this is my placebo distribution. This is null. All right, the placebo distribution. This is the alternative distribution. All right. So I have the null hypothesis and the alternative hypothesis, where the difference between placebo and test drug is delta. All right, it's delta. Remember, I said I want delta to be positive. So that delta is standardized already divided by sigma. All right. So from 0 to delta, and on top of 0 is the placebo distribution. On top of the delta is the alternative distribution. So we believe the drug can deliver this kind of efficacy. And here is a critical point of Z alpha. So in other words, from here to here is Z alpha. If your alpha is 0.025, this is 1.96. Okay. And so under null to the right of Z alpha is alpha. All right. So 0.025. Okay. Now, to the left of this line, this part of the probability, under alternative, this beta, right? So under null, from this point to the right is alpha. Under alternative, from here to the left is beta. And from here to the right under the alternative hypothesis is power. That's 1 minus beta. So that's how we understand this basic construct. Okay. Now, So I have this Z alpha and Z beta. For Z alpha plus Z beta, you got this delta here, right? Now, if your sample size is small, then the two distributions are pretty flat, right? They're very flat distributions. And of course, your area to the left of Z alpha and to the right of Z alpha to the left of Z alpha, it's going to be a large area. All right. So now what you do, you increase sample size. When you increase sample size, your distribution looks steeper and steeper and steeper. Once you reach this steepness, your Z alpha from here to here is alpha, from here to here is beta, and you stop. And that's your sample size. So that's how we understand these two curves. Okay, so when we calculate sample size based on the assumption of alpha, beta, delta, sigma, that's the sample size reflect this design. All right. So now this is what we had with the design with a given sample size. Now I sit down with the team. I said, after unblinding, you're going to have the study results on the primary efficacy endpoint. You're going to see something probably to the right of delta. I mean, if you see such a nice efficacy response, everybody's going to be happy. Says, hey, look, it's statistically significant, clinically meaningful. It's a very good drug. So the decision is clear, right? See, you see some data to the right of delta, piece of cake, no brainer. You make a go decision. All right, that's easy. Now, if you see something to the left of zero, people are going to be disappointed since, well, the drug doesn't work. It's even worse than placebo. Fine, it's an easy decision. You make a clear decision. So where the problem come from, if you observe something here, it's clinically not meaningful, but statistically significant. Should you make a go decision or no go decision? And of course, different people have different opinion. Okay, so what are the consequences? If you make a go decision, your alpha is not compromised. All right, you make a go decision, statistically significant, even though it's not clinically meaningful, but your alpha is protected. But if you're making a no-go decision, your beta is inflated. If you observe something in this area, your beta become zero point 0.5 if you make a no-go decision, right? Because your beta, your type 2 probably was from this line to the left, and you observe something here, make a no-go decision, your beta is inflated. Okay. But these are not hard decisions. Okay. The hard decision came from here. In most cases, in my experience, we observe something here and the team want to make a go decision. I said, wait a minute, it's not clinically meaningful, it's not statistically significant. How can we make a go decision? And here's what they tell me. Naitee, remember when you designed the study that delta has been artificially inflated. Naitee, you said you want to get the sample size feasible, so you have to have a large delta. A drug deliver a delta like this much is a wonderful drug. That delta on the right is fake. Well, I take it. Fine. Okay. And then I ask the next question. What if you observe something here? Oh, no good. The drug doesn't work. It's so close to placebo. So if I move back and forth, sooner or later I can get a point everybody agrees. So below that point, it's no go decision. I say, fine, if we agree with that, let's document it. Below, if we observe something below this value, it's a no go decision. So I call that LRV, lower reference value. And I call the delta as target value. So now the decision is a little bit easier. All right. If you observe something to the right of delta, it's a go decision. Fine. If you observe something lower than LRV, it's no go decision. Now, between LRV and target value delta, that area is gray area or the area of inconclusiveness. You cannot make a decision. All right. So in reality, after blind is broken, everybody is going to see the data and everybody has something to say, and they're going to look at the totality of the data. They're no longer just looking at primary efficacy endpoint. They're going to look at secondary efficacy endpoint. They're going to look at safety endpoint. They're going to look at whatever they have to make the decision. But if we have this discussion before unblinding, at least the team knows if you observe something below this point, it's not likely you can save this drug, right? But if you observe something over this range between LRV and delta, at least you can look at the totality of the data and you can make a better decision. So this is the communication tool I try to propose. Okay. So now these are just right out what I just said. Now let's move to dose ranging. So I hope with these few slides, I can convince you POC is very, very, very difficult. And now, if the concept is proven, we're moving to dose ranging, the next big part of phase 2 trials. Okay. Dose ranging, of course, parallel studies, placebo controlled, duration of treatment limited by MOTOX coverage. Well, I had to deal with this in many of the phase 2 trials. Sometimes by the time we design the phase 2, the tox coverage is only 2 weeks. All right. So they did probably one or two different species, but follow only 2 weeks. And then, sorry, your clinical trial has to be limited to 2 weeks. But if they have 4-week tox coverage, we can do a 4-week trial. So phase 2 usually short term. You got 4 weeks, 2 week, 4 week, 8 week, 12 week. The longest phase 2 is half a year. So in my experience, of course, from chronic disease point of view, 4 to 12 weeks is long enough. Could be long enough for a phase 2 decision. All right. Many doses of test drug. So what would be a typical dose ranging trial? High dose, medium dose, low dose plus placebo. All right. Objective is to explore a range of efficacious doses. The key word here is to explore. We're not trying to confirm any dose that works or not. We're not trying to make a decision. Although people says Naitee wasn't make a decision, we want to make a decision. No, dose ranging by itself, by nature, is exploratory. We're just explore the range of the doses that is both efficacious and safe, and we can only explore. Remember, minimum effect dose is mission impossible. So minimum effect dose, I just said. Oh, by the way, yeah. When I talk about the difficulty of minimum effect dose, when I say minimum effect dose is mission impossible, is that if you understand how difficult the POC is, all right, the POC, we have to go through this discussion and look at inflated delta, all these problems. But POC is only at MTD at the highest dose. All right, when you do a dose ranging study, have high, medium, low dose, you're gonna have every dose. You're gonna have this POC discussion, right? So from a dose ranging design, there are multiple test doses. When each dose is compared to placebo, there's a POC discussion. All right. And then we have to identify which dose is efficacious, which is not. Among the efficacious doses, we'll try to find the minimum. That's why minimum effect dose is mission impossible. All right. Your sample size is limited to begin with. And you try to come up with some kind of decision about minimum effect dose. Forget it. No way. But what we try to do is to try to understand the shape of the efficacy dose response, try to look at the safety of these doses we have studied. That's the best we can do. All right, what does it mean by a dose range for a given study design? All right, suppose design A, I got placebo 20, 40, 80. In design B, I have placebo 0.1, 1, and 10. Which design has a wider range? All right, is that A or B? Wait a minute, Naitee, you have to define what is dose range. So dose range is defined for the same design. It's a high dose divided by the low dose. It's not subtract. So if you look at subtraction, A has a range of 60 milligram. B has a range of 9.9. Looks like A is wider, but in fact, it's not. Subtract is divide. Dose range for a given study is defined as the high dose divided by the low dose. All right. Design A has a range of 4, design B has 100. Why dividing? Okay. By looking at these two studies, these are two different drugs. Drug A may have an MTD of 100. Drug B may have an MTD of 10 milligram. Okay. So when you design a dose ranging trial, you want to have a wider range. Wait a minute, your upper limit is bounded by MTD. So if you want to have a wider range, the best way is to move the lower range, the lower dose to be lower. So the lower the low dose goes to, the wider the dose range is. It's not the higher the upper dose. Higher the upper dose is bounded by MTD. So you can only look at how low you can go. All right. How do we design dose ranging studies? Of course, we have to consider the number of doses. Do we need active control? The problem is really a dose spacing problem. And of course, we have to use the clinical endpoints and length of study. All these considerations has to be put in together when we design a phase 2 dose ranging trial. All right. Now, traditionally, we have dose ranging and POC separate. All right. So you have proof of concept and dose ranging. But nowadays, a lot of companies just combine them. So remember, I probably forgot to say this. All right, this set of slides I call it Dose Stanford is because in 2017, Stanford University invited me to give a short course about those phase 2 dose finding kind of stuff. So I took my collaborator, Dr. Naitee Ting. So Naitee did the second half of the short course. I did the first half of the short course. So in the first half of the short course, I did the traditional way of doing it, POC then dose ranging. And for Chi-Chi's slides, she tried to combine POC and dose ranging into one study. So that goes to her study. So I would say why POC and dose ranging separate traditionally, because we're not sure if the test drug works, right? If you start with a dose ranging trial, you have high, medium, low dose plus placebo. It's more investment, but not even knowing whether the drug works. All right. And sometimes there's formulation or dose strength limitations and extrapolation from PD markers of clinical efficacy and investment cost and possible ethical concerns. So if you don't know the drug works, how can you recruit a lot of patients? So you start with a POC, and then after concept is proven, you know you have a drug, you move down to dose ranging. All right. But this POC decision is very critical. All right. The POC decision would affect the drug formulation. All right. Because before POC, you're not sure whether you're going to phase 3 or not. But now you have to go to phase 3, and sometimes there's possibility you're going to market the drug. So you have to have the drug formulation for phase 3 for post-marketing. Do you want to order a large quantity of raw material? If concept is not proven, I'm done. I don't need to. Um, I I don't need to order a huge amount of raw materials to prepare phase 3 studies. But if concept is proven, I will need to start ordering a large quantity of raw materials. And also, I mentioned about toxicity coverage. All right, if you engage in phase 3, you better have long-term tox studies like 26 weeks or 52 weeks kind of task coverage to engage in phase 3. But before proof of concept, I don't have to worry about all these investment. But once concept is proven, I have to make these decisions. So a clear go no go decision from POC is very critical. I have to make this decision. Delaying this decision is this period of time I call inconclusiveness. Remember, there's an inconclusive zone. Greater than delta is go decision. Less than LRV is no go decision. In the middle is inconclusive zone. Inconclusive zone, that's from that point of view. But from drug development point of view, you have to consider the time scale. There's a time of inconclusiveness. And can we afford such a long time of not being able to make a go no go decision? All right. Risk of inconclusiveness. So typically, the clinical trial process, you design, conduct, unblind results, but from results, can you make a clear go, no go decision? The question is to go or not to go. This decision has to be made. There's nothing in between. You have to make that decision. This molecule is in front of you, whether you want to continue to develop or you stop developing it. And the clock is ticking because the patent life is running out. You have to make a decision. And the other thing is that the team finally can make only one decision, whether it's go or no go. So they can make only one mistake. So if they make a go decision, there's no beta issue, right? It's an alpha issue. There's an alpha probability they make a wrong decision. If they make a no go decision, there's a beta issue. There's no alpha issue. All right. This decision has to be made. Delay this decision impact formulation, order of raw materials, tox studies. Inconclusiveness happens between study results and decision. So from here to here, that's inconclusiveness. All right. After results are ready, there's very little statistician can do. The critical time for statistician to help the team is at the design stage. So clearly communicate type 1, type 2 risk and define go no go criteria. That's very important. That's where we make this most important contribution. So before unblinding, we're the brain of the team. But after unblinding, I became the pair of hands. Just do this, do that. So now we move into a dose ranging. All right. So people are used to this kind of dose response curve. All right. So as dose increases, the efficacy increases. That looks good, right? Now imagine if you think about the petri dish kind of situation. If you keep increase concentration and there's no response, you don't have a drug, right? Same thing. All right. In clinical trial, if increase in dose you see no response, you don't have a drug, right? But if you have a drug, if POC, the curve, excuse me, has to increase somewhere. Suppose I have a very safe drug here. If my MTD, maximum tolerable dose, is 1,000 milligram, 1 gram, 1,000 milligram, that's my MTD. Very, very safe drug. Now, I try to design a dose ranging trial to capture this curve, right? So if I know the curve is like this, I can do my high dose here, medium dose here, low dose here, and placebo. The problem is I don't know where this curve is about because when you design dose ranging trial, you're trying to use these doses to capture the steepest increase part of the dose response curve. But unfortunately, I don't know the steepest increase part is between zero point zero 0.01 milligram and zero point 0.1 milligram, or between zero point 0.1 and 1, or between 1 and 10, or 10 and 100, 100 and 1,000. I have no idea. So that's a real challenge in designing the first dose ranging trial. At MTD, the drug works, and I'm making another assumption, which is we call monotonicity assumption, says the efficacy can only increase when your dose increases. In other words, this curve doesn't go down. So I'm making some assumptions. I have to assume the MTD was guessed correctly, although there's no guarantee. And from practical experience, we see both cases, MTD being underestimated, being overestimated. And the next thing is that I have to assume the monotonic dose response relationship. And even with those two assumptions, there's infinite possible number of curves in this placebo to MTD range. All right, so that's a huge challenge. Another problem is that in practice, we never see a curve like this because you design a study with some discrete doses. You only get to see the responses at certain doses you study. So you never get to see a continuous curve. Another problem is that there's individual curves. Well, this curve might be Schilling's curve, this curve might be my curve, this curve might be Dirk's curve. Everybody have your own dose response curve. And in clinical trial, we can only study an average dose response or population dose response curve. We don't get to see the individual dose response curve. And also is even though I assume efficacy is monotonic, and I've been challenged in many situations, but people generally believe the toxicity curve is monotonic. So as those increase, your efficacy increase, your toxicity increase. All right. Suppose here's 0%, here's 100%. All right. Now, if you take 50% here, So this 50% line intersect with the efficacy curve, and that curve, that dose we call it ED50, the dose that delivers 50% of efficacy. And you can continue to the toxicity curve, and that dose is what we call TD50, the dose that delivers 50% of toxicity. Okay. Now, people believe that between ED50 and TD50 is what they call a therapeutic window or therapeutic index. I don't think so. I think ED50 may be okay, but I think I'll probably stop at TD10 or TD20. Okay, so I probably won't go beyond TD20. So this would be the area you can dose in a way that is both efficacious and safe. Okay. So what would be an ideal drug? An ideal drug where the efficacy goes up like this. All right. At very low dose, you got 100% efficacy. But the safety curve goes like this. Okay. It's almost flat. You keep on increasing doses. The drug is very safe. And then you get a very wide therapeutic window or therapeutic index. So what the advantage? For a wide therapeutic window, you can just take one dose, all right? So just slightly to the just around here. You can just take one dose such that the same dose could work for 80-year-old women with very small body frame. And the same dose works for a young macho guy, maybe 100 kilogram body weight or something like that. All right. So you have one size fits all. You have one dose that covers. So that's an ideal dose response curve, right? But most curves are like this. All right. So you have to dose very carefully. Now, we talk about chemotherapy. Chemotherapy, the two curves are almost touching each other because the efficacy in the chemotherapy came from the ability the drug could kill tumor cells. If it kills tumor cells, it must be toxic to your ordinary cells. So these are the different challenges of trying to find out this um, therapeutic window or therapeutic index such that you can dose these patients that for these patients is relatively safe and is efficacious. So, like I said, in reality, you never see those curves. The best case scenario, you probably see this. You look at the efficacy response, and uh, you have so many doses, and you see a clear dose response relationship. But in practice, what you see is a response for placebo is between 0 and 100%. Every dose is between 0 and 100%. You probably have 3 or 4 or 5 doses. And the distribution are all over the place. And we plot the mean or median, you see a tiny slope. And you say, hey, there's the dose response. Okay, so that's what we're dealing with because in clinical trials, human to human differences are huge. You try to tease out the signal out of the noise, it's not that easy. Um, I think I'm almost running out of time. Let me just get into a case study, okay? All right, before that case study, I usually ask people, I say, low dose, medium dose, high dose. Do you see a dose response? Well, 90, it looks like it's dose response. But if I tell you placebo is here, you don't even have a drug. So placebo is absolutely critical in dose ranging trials. We have to have placebo control. Now, what about this drug? All right. Now, I have placebo, low, medium, high, and there's active control. Active control is the dose. The drug is a different drug. It's already worked. It's already marketed. So we take that drug as active control. And clearly, the test drug doesn't work. All right. So when I show this to many of the drug developers, I would ask them, how do you think? And most of them feel sad because the drug doesn't work. Because by this time, remember, you've done the preclinical discovery, development, phase one, POC, and now dose ranging, and you end up with such a result. People are going to be disappointed. I say, no. I say, this is a good study that failed a bad drug. The study delivers a clear no-go decision. All right. A failed study look like this. All right. you cannot differentiate test drug from, placebo from active control, or you cannot differentiate. So there are two possibilities. One possibility is that in this study, the placebo response is extraordinarily high, so the drug still works, but the placebo is so high, you cannot make that decision. Another possibility is that in this study, the active control just worked very poorly. So this is an example of inconclusiveness. You cannot make decision. Okay. Now here's a case study. All right. I joined Pfizer in 1987. Starting in 1988, I worked on this trial. I worked on this drug, and this is the first dose ranging trial. Now, the y-axis is pain scale, and we're looking at pain reduction. It's a 4-week trial looking at pain reduction. And the pain scale we set was 0 to 31. 31 is very painful, and 0 is no pain. And you look at these results, clearly the drug works, okay? Placebo still improved maybe about 5 points, and the test drug improved about 20-some points. It's very impressive. So the first conclusion is the drug works. The next thing you see is that, well, I didn't see a dose response. So it could be that the 80 milligrams is too high, right? So we designed the next study, 40, 80, and 120. Okay. And then we went to phase 3. After study 2, the phase 3 study started with 120 milligram. And in the phase 2 meeting, FDA questioned about dose. And we designed the third dose finding study, look at 2.5, 10, and 40. Okay. So the efficacy range is around 20 to 40. All right. The consequence, we had to redesign phase 3 studies with 20 and 40 milligram. It took us 3 studies to find dose ranging study, the right dose response. The large scale study was 100 milligram, cannot be used for registration, and filing was delayed by many years. It's a very huge consequence. I analyzed this study. I still remember that day. It was 1993. I was in my office, plugging the computer and run the SAS, and I got those results. I was completely shocked. Almost everybody saw the results, was surprised. You know, in the first study, you did 80, 120, 160. In the second study, you did 40, 80, 120. You know, it's easy for me to show these 3 bar charts. The entire time frame could last 5 or 6 years. And it was very important drug for Pfizer. And at that time, I was working on this drug, and I saw these results. I was completely surprised. And this drug motivated me into this field of dose finding, dose ranging. So I want to stop here, and we can sort of open up for Q&A.
Thank you, Naitee, for the great presentation. So now we can move into Q&A. And if anyone has questions, there are several options. One is you can enter in the chat window. And the other way to do it is that when you move the mouse next to your name, you will see that there's a hand. You can raise hand, then we can unmute you. Oh, if needed, I don't know whether you can unmute yourself, but I don't think so. But you can raise hands. I do see a few hands raised, and Dirk can help unmute the ones with raised hands. So, Dirk?
I just unmuted Barrage.
Hey, can you guys hear me?
Yes. Hey, so great presentation. Yeah, like this is one of the best presentations on this topic I've heard, and I've heard several. So really like clarifying presentation. Thank you. So my question is about the slides where you presented about the two distributions that you were talking about, the LRV, the lower reference value.
Yeah.
Yeah. So like, uh, I am a little bit confused about like what is the like, is there a mathematical or a statistical way this LRV is determined, or is it a fully ad hoc decision made based on the expertise of the clinicians and the statisticians involved?
Excellent question. I really appreciate this question because this really touches on the point about a pharmaceutical statistician or clinical statistician, what we should do. All right. Yeah, our education on mathematics and statistics. But what are we doing here? We're developing drugs, we're helping patients. So the objective of our job, the objective of FDA's job, the objective of NIH job, of everybody is about public health, it's about patient care. So we have to understand our role. Our role in this industry is to help patients. So whenever we see a problem, we tend to think from mathematics and statistics as our training. But for our job, the job requires us to think from the patient's point of view. Now, if you think from the patient's point of view, I cannot directly help the patient, but I can help the physician to help the patients. So that LRV, instead of ad hoc, I would say is a result of discussion, of mutual understanding. As a statistician, remember I try to play as a thinker. We have to let people understand our value. The only way people can understand us is we can talk their language. So this is a job you can forget about mathematics. When you think about statistics, you should think about the statistical principle, not statistical methods, because statistical methods over, I wouldn't say a percentage, is piece of trash. It's totally useless. It's really, do you understand the problem? Or do I understand the problem? I really appreciate this question because this question reflects, you can tell I'm trying to explain the concept, trying to understand the question, and I'm not dwelling on any methodology or any statistics because the statistics and methodology is just try to get the data analyzed and things like that. So I would not call that decision being ad hoc. I would call that decision being a compromise. We are in a real world setting. Remember, the most people we work with are not statisticians. They don't know what statistics are thinking. They thought we are mathematical genius, but we don't know how to communicate. And that's where we need to put our effort on, trying to learn to communicate, try to totally forget mathematics. It's hurting us more than helping us. Thank you.
Okay, let's go to the chat. There's a question from Shuhao Zhu. One drawback in dose ranging is the challenge of randomization for a larger sample size to delay drug development. Could you comment on how it can be overcome?
Well, that's again very good question because people tend to think of overcome some problems. But before we try to overcome it, do we really understand the problem? Okay. The I don't have to do a large sample size. I can do a regular sample size with increased delta, alpha, and beta. All right. And my objective is not trying to overcome the problem. My objective is trying to understand the problem. So for a given sample size, we can come up with some data. With these data, can we help make some more informative decision? Yes, giving me an opportunity, I would like to go with a large sample size, but that large sample size has to be limited by practical considerations, including the ease of recruitment, the budget, the practicality. So I love my career as a clinical statistician, being that I'm in the real world situation. I'm always compromising with the feasibility that's around me. So I not try to overcome anything. I try to understand what I'm doing.
Okay, let's go to the verbal questions. We have Georgia Chow. You're on.
Georgia, unmute yourself, Georgia.
You're muted.
Unmute yourself, Georgia.
Okay, try. Just a minute.
Still muted. So maybe you do need to click in the bottom part of the screen. There's a mute and mute. You need to unmute yourself.
Okay, let's go on to the next one, and then maybe Georgia can try again. So we have Marlena. Marlena, you're on.
For the hand raised, but it was an excellent presentation. I really enjoyed it.
Thank you.
Okay, let's go to the written questions. Jim Bolognese writes, can you mention your thoughts on using Bayesian prior posterior probabilities instead of alpha and beta?
Good question again. I was trained as a frequentist. Throughout my life, there's never a day I believe in Bayesian. It's just my own opinion.
Okay, next question from Marzana Chowdhury. This is a great presentation. Given that there is no guideline of how number of doses we should explore in phase 2, what is the minimum we should do? I believe it should be more than 2. Thank you.
Thank you for the question. If you look at my publications, I do have publications on number of doses. My recommendation is 4 to 5, at least 4 to 5. You can look at my publications.
Okay, and then we have Xu Hao Zhu as a follow-up question. Thank you. Could you also comment on the major bottlenecks in existing methodology for phase 2 trial design?
Oh, okay, sure. The major bottleneck is people believe model. The model hurts the drugs. Model hurts the companies in huge numbers. Companies after companies are losing billions of dollars because people believe in dose response models. There's no such a thing as a dose response model. It's a big, it's a, how should I say this? Well, the danger or the huge risk of believing model is that if you think of a model, you tend to believe the model. You try to look for the optimal dose for that given model, and when you design, you get a narrow range. You didn't get a wide enough range. When I say wide enough range, you want the lower dose to be low enough. And they say, what if the low dose doesn't work? That's precisely what we want to find. If you find the dose that's so low it doesn't work, the dose above that works, people believing that's a mean effect dose. Remember, mean effect dose is mission impossible. It's never well defined. But if you can find the dose low enough, it doesn't work, then that'll help you anchor where lower end of the dose range should be. So never believing any dosage models. All models are wrong. Some are useful, but in dose response, they're totally useless. Never believing any models.
Okay, let's try Georgia Chow again. I think maybe we can hear you. I'm trying to unmute you again.
Georgia?
Okay, well, that didn't work. I'm sorry. So we have now a question from Mia Teixeira. Can you explain a bit more why you don't believe in Bayesian?
Oh, okay. Because when I talk about model came from assumption. In my experience in drug development, the one thing that hurts every pharmaceutical company most is assumption. All right. I see mistakes after mistakes, disasters after disasters. Everything when that happens, they say, oh, it is not because we didn't know. It's because we thought we knew. That prior really hurts us so much. There's no such a thing as a prior belief. Almost everything we believed was wrong turned out to be wrong. That's huge disasters. It's bloody experience of huge mistakes. Mistake after mistake is that we thought we knew, and we actually don't know anything. Flexible dosing is a totally different question. So let's talk about dose titration. I didn't have time to talk about dose titration. Okay. So let me design the titration study. Okay. Test drug against placebo. All right. So the titration is 10 milligram to 20 milligram. All right. So I have placebo, 10 milligram placebo. So I start out everybody randomized to test drug or placebo at 10 milligram. And if the patient doesn't have efficacy, I can titrate up to 20 milligram. All right. So 10, 20 titration. So placebo, I have the 2 tablets being 20 milligram placebo. All right. Now, at 20, if you see adverse events, you can down titrate to 10. Okay. So we have such a design. And then the clinician says, Naitee, now the study's over. I want to see a dose response. I say, I don't have analysis data for you. I know 10 milligram is more efficacious than 20. And 20 is safer than 10. And he said, how is so? All right, think. For a patient does not respond to the test drug, at 10 milligram, the patient does not respond. So you titrate to 20, the patient's still not responding, right? But for patients responding at 10, stay at 10. So of course, 10 milligram looks more efficacious than 20. And for patients at 20, if it's not safe, you down titrate to 10. But if it's safe, you stay at 20, right? Of course, 20 looks safer than 10. So titration design is a totally different disaster.
Okay, we have a question from Shailing Ron. What are your thoughts on flexible dosing?
That's what I talk about.
That's what I was.
Okay.
Actually, 19 was just discussed that, but 19, I just want to follow up. Actually, in fact, FDA does accept flexible dosing in some therapeutic areas, like I mean, of course, we don't need to name the therapeutic areas, but the
therapeutic area matters. Therapeutic area matters. Not only therapeutic area matters, disease matters, drug matters, everything matters.
Right. So what on your the trail of thoughts that you just mentioned? So the higher dose, because it's safe, so we stay there. It does. So that means the higher the dose, the better the efficacy, right?
That's the assumption of monotonic assumption. So the monotonic assumption for efficacy, not everybody believe it, but everybody believe toxicity is monotonic.
Right. So the question is, so the titration design is the only it's the design to support a flexible dosing.
Well, so that design answers the question. It answers a very good question whether the drug works or not. Okay, but it didn't tell you the dose response. And titration design is very good in practice because in practicing physician, that's how they treat patients. They start with a low dose. If it doesn't work, they increase to higher dose. Doesn't work, they increase a higher dose. So the titration design mimic the real world application, but it does not answer dose response questions.
So in your experience, if we have a titration design, is a dose ranging design still needed?
Well, depending on the question you want to ask. All right. Now, there are forced titration, which is very different. All right. So I worked on a drug called Cardiora. All right. Daxicitine. Daxicitine is an alpha blocker. All right. So the long-term maintenance dose is like 8 milligram or 16 milligram. But we had to do a forced titration. We start with 1 milligram. 1 milligram for 1 week, and then 2 milligram for 1 week, then 4 milligram for 1 week, and then 8 milligram, or you can titrate up to 16 milligram. Why? Because that's a forced titration. Because if you start the patient at 8 milligram, the blood pressure is going to lower too much. The patient cannot stand up. So you have to do a forced titration. So that's very different. So for titration design, you can answer a question about regimen, not a dose.
Okay. Okay. Yeah. Thank you.
Yeah.
Okay, well, first of all, there's a robust discussion in the chat about Bayesian priors, so you all can read that at your leisure. Anyway, Alex Sturdlove writes, thank you for a great talk. Can you comment on any experience of partnerships with pharmacometricians on dose ranging finding designs? Also, you mentioned we should not believe in models. How about model-informed drug discovery and development, MID3, that is in the spotlight of modern drug development?
I completely respect model-informed discovery, but development, it may not be of dose ranging development. Anything up to dose ranging development, don't believe in any models. All models wrong, and all dose ranging models are useless, completely useless. Okay. Now, the work with pharmacometricians, actually, I learned a lot from them. But when we communicate them, we have to, it's kind of mutual education, but we have to understand the roles of pharmacometrician and statistician are fundamentally different. I respect pharmacometricians because the way they look at data, they try to summarize the data from the same patient, like AUC, Cmax, Tmax, into a single point. And they try to make inferences on these individual patients. And I learned so much from them. But statistician is different. Statistician is looking at a group of subjects. We're looking at, there's a population. We take a sample from the population and we try to make inference back to the population. That's our training. We're looking at the population. We're looking at a group of subjects. And that view is very different. So how we collaborate, the pharmacometrician give us a single point, and we can take the mean or geometric mean to help them understand the population effect. So in other words, my training doesn't give me the education to summarize the data from the same subject into a single point, but they have that training. So that's how we collaborate.
Okay, well, there is one more question about Bayesian. So Yue Bao says, I am a Bayesian student, which makes me wonder if the priors lead to bloody experiences, where are the places in drug development for Bayesian statisticians?
I respect them, but I've never worked with them. I'm a non-Bayesian. I'm 100% frequentist. By the way, I want to emphasize everything I said reflects my personal opinion. And I want to make clear that my opinion is not trying to convince you to take my opinion, but to encourage you to formulate your opinion. It's be best your opinion is different from mine. Because for people who just new to the industry, they don't know much. Of course, they don't have opinion. But if you're in the industry for 5 years or 10 years, you still don't have an opinion. That means you did not learn anything. If you learn something, you must have your opinion. And I never said my opinion is correct, but I want to give you clearly that's my opinion.
So, Naitee, I have another question. So, it's phase 3. Is it always prefer to just pick one dose go into phase 3, or is there any thinking on like taking 2 doses into phase 3?
I have a couple collaborators. We're now working on manuscript. I'm trying to discourage just one dose, but companies want to use one dose because it saves the investment. But one dose is very dangerous, very risky. Here's why. All right, it's well known that phase 3 dilute phase 2 efficacy. There are many reasons why they're diluting. So the first reason would be in the inclusion and exclusion criteria. Phase 2, you don't know whether the drug works or not. So from phase 2, you want to give your test drug the best opportunity to show its efficacy. So you want patients doesn't have very severe disease condition because if it's too severe, no drug can help. You don't want their situation to be too mild because if it's too mild, it's not differentiated from placebo. So you want to have a certain inclusion criteria to give the drug the best opportunity to demonstrate its efficacy. Phase 3 is different. Phase 3, you want to mimic the market populations. You want to relax the inclusion and exclusion criteria. Of course, it turned out your phase 3 efficacy will be less than your phase 2 efficacy. So because this well-known phenomenon, people tend to have their phase 3 dose too high. Now, those too high, there's one potential problem, which we actually saw that, is what I call late developed adverse events. So your phase 1, phase 2 is probably at most maybe 3 months or 6 months. Phase 3, so we engage in phase 3, we're committed. Phase 1, phase 2 looks great. The drug goes to phase 3. At 9 months, there's a new AE. We never saw it before. And that could mean the drug accumulate over time in the human body. And 9 months is a new AE. There's nothing you can do about it. You cannot stop your phase 3 because you already invested in the whole phase 3, the large program. You can only observe and pray. Hopefully, there's no more events. And after 12 months, at 13 months, 14 months, there's still AE pop up. And at this point in time, what can you do? There's not much you can do. So I would try to encourage always have a lower dose. The high dose is good in efficacy, but once there's a late developed AE, the low dose may save your day. So that's my recommendation.
Okay. Yeah. Thank you. So I don't know if there's anyone has additional questions. I don't see anything. In fact, I have one more question. I think a lot of people might have the same question. What's your thoughts on combining phase 2 and phase 3? Because there are
I would never
currently
do
a lot
that.
of
I would never do that for chronic
efficiency.
disease. I would never do that for chronic disease because FDA gave us a huge favor, which is called end of phase 2 meeting. If you do a combined phase 2, phase 3, you're wasting that good opportunity because the end of phase 2 meeting is not only about reviewing your phase 2 results, but FDA will give you recommendation on phase 3 design. Is the endpoint correct? Is the timelines correct? Is the design acceptable? So don't waste that opportunity. Do separate phase 2 and phase 3. Another thing is that phase 2 is the last chance you kill a drug. Okay? Phase 3 is a huge commitment. Don't just easily agree with his phase 3 investment. It's very risky. Remember back in 2016, Eli Lilly has a failed phase 3 for Alzheimer's disease. They have to lay off 3,000 people. Is that a worthwhile gamble? Do not do phase 2, phase 3 combined.
Okay. Yeah. Thank you for your insight, Naitee. Any other questions from the audience?
I don't think so.
So if not, I think we want to first thank you. Big thanks to Naitee for such informative and also very presentation and also very thorough discussion on this topic. I hope everybody gains some insights into this topic, regardless of which phase of the clinical trial you are working in. And if you have any questions, please feel free to reach out to the organizers. If you have question for 19, you can also email us. We will help connect you with 19 too. And we will post the slides in the ASA NJ chapter website, but please give us a few days to do that. We normally don't operate in the lightning speed, unfortunately, but so give us a few days. So we will post that, and also we do have a recording which will only be available upon request if you do want to hear the recording again. All right, so that I think that's it for today, and I want to thank Naitee again, and also thank you everyone for attending, and thanks to all the organizers, especially Doug, for hosting this.
Thank you, everybody. Thank you for your help.
Thank you. Thank you, Naitee. Thank you.
Yeah, thank you. Okay,
Bye.
bye.
Bye.